Students, in today's class we will study about the chromosome, its composition, its different types, and then the composition of DNA. In lower classes you have studied about the cell. It is animal cell or plant cell. The basic components in the two types of cells or the living cells. Suppose this is animal cell, this is plant cell. So whatever may the type of cell, this part is known as cell membrane. Cell membrane. Here this is also. Although in plant cell there is an external layer also made up of cellulose, that is called cell wall. Cell wall is freely permeable and rigid in nature. So this is cell wall. Now then, inside the cell membrane there is the protoplasm. In the animal cell, in the center almost, there is a round body called nucleus, which has condensed structure called nucleolus. Nucleolus. This is nucleus. And inside it there are many fine thread-like structures which are called chromatins. In plant cell, since there is large vacuole inside, this is vacuole in which the cell sap is contained, and then its cytoplasmic organelles are on the peripheral side. So nucleus will also be on the peripheral side. Suppose this is the nucleolus, chromatin fibers. So this is nucleus. Then there are many cell organelles, like this is the mitochondria. Now there are many flattened sac-like structures, that is Golgi body or Golgi apparatus, Golgi body. There are further more other structures like lysosome and other structures. In a plant cell there are, suppose, mitochondria, not lysosome, it is found in only in animal cell, and then there are small structures in large quantity, that is, suppose, chloroplasts. So this is the nucleus. You see here that inside the nucleus there are many long, highly coiled thread-like structures called chromatin fibers. These chromatin fibers at the time of cell division coil, condenses, shorten and become visible as chromosome. So we use the term chromosome generally at the time of cell division, but in normal condition it is called chromatin fibers. Now, chromosomes in animal cell, it was Walter Fleming who studied chromosome. So in 1882, Walter Fleming studied chromosome in animal cell. Now you see here, students, in the chromosome, which will be term generally used at the time of cell division when it is highly condensed chromatin fiber. So if you say what is chromosome, chromosome, then it is highly coiled. We also say supercoiled. Highly coiled and condensed, coiled and condensed chromatin fibers. Chromatin fibers are called chromosomes. Are called chromosomes. Chromosomes. Now, so this is chromosome. Now, in chromosome, if we will study its structure, then it has highly coiled condensed structure. Highly coiled condensed structure. Now, there are two threads, one and the two. Second, these are called chromatids. Chromatids. Now, the two chromatids of a same chromosome, that is these two, are called sister chromatids. This is the centromere. Centromere. So, if we study chromatid, this is the thread-like structure. Two threads or strands in a chromosome are called chromatids. Now the centromere. Centromere is the point of attachment of two chromatids. The centromere also provides the point of attachment of spindle fibers at the time of cell division. That is, centromere is spindle fibers join at centromere, and when they contract, then longitudinal splitting of chromosome take place. So, centromere. Now, there are 2 points related to centromere. Centromere. First, point of attachment. Point of attachment of two chromatids. Chromatids. And second, it provides point of attachment of a spindle fiber. That is, provides point of attachment of spindle fibers. Spindle fibers at the time of cell division. At the time of cell division. So, this was the general points related to chromosome. Now, in the cell, there was another structure called centrosome. In case of animal cell, centrosome, animal cell, it is not present in plant cell, animal cell. Centrosome. It is the clear space in the cytoplasm near the nucleus, which contains two bundle-like structures called centrioles. And the centrioles give rise to astral rays and spindle fibers. So, centrosome in animal cell initiate cell division. So, if the question is asked that what is the function of centrosome, then we say it initiates cell division by, that is, it divides into two centrioles, which are bundle-like structures in the centrosome. So, this question is asked frequently. Name the cell organelle that initiates cell division. You will not write centriole, but you will write centrosome. Now, here next we have to study about the composition of chromosome. Composition. composition. Now, the chromosome or chromatid, it consists of two chemical substances. First, histone protein. We represent it in the form of ball-like structures, small units, small proteins. And second, DNA strands. Now, if we draw the condensed structure of a chromosome, now as you know, it is highly coiled structure. If this is unrevealed, you will find that it is similar to the wire of receiver of a telephone. Now, when it is further unrevealed, you will observe that there are about 8 small ball-like structures. These ball-like structures are histone, and 8 ball-like structure is then surrounded by DNA strands, which further divide into 2 strands. So these 8 histone proteins are winded by DNA strand, and then this is together known as nucleosome. So now you see, that is the unit of chromosome or chromatid is nucleosome, or we can say the repeating unit of chromatid is nucleosome. Now again, nucleosome or chromatid consists of 2 chemical substances. Histone protein. Question is asked, name the protein present in chromosome. It is histone protein. So in a nucleosome there are 8 histone protein molecules winded by DNA strand, and this unit is known as nucleosome. So this was about the composition of DNA, composition of chromosome. Now, one thing we will study here, that is about classification of chromosome. Although there are many methods to classify, but for simplicity we will study here for knowledge, because we will study it later on, terms will be required. The first basis to classify is on the basis of function. We can divide it into 2 parts. That is, first part is autosome. Now in human being it is 22 pairs. That is 44 ह्यूमन बीइंग के लिए. डिफरेंट एनिमल्स में अलग अलग पेयर ऑफ नंबर ऑफ क्रोमोसोम्स होता है. तो एनिमल्स ह्यूमन बीइंग में 23 पेयर ऑफ क्रोमोसोम्स होता है, आउट ऑफ विच 22 पेयर्स, दैट इज 44 आर कॉल्ड ऑटोसोम. नाउ व्हाट आर दीज? They carry genes for general body features, or somatic characters. somatic characters. Means you see that this will be similar, same, with slight genetic variation in males as well as females. That is, the male and female, like in human being, the general body design is the same. The different organs and their functions, behavior are the same. But there is another one pair of chromosome is called sex chromosome. Sex chromosome. Now the sex chromosome is one pair. We say 23rd pair, 23rd pair, one pair. And this sex chromosome carry genes for sexual characters, that is for development of sex organs, development of gametes, formation of gametes, secondary sexual characters and so on. Carry genes for sexual characters. Hence, the males and females will have different types of chromosomes. Like you know, in male, it is XY type. That is also called heterotypic. Heterotypic. Hetero means different. Means this pair will have two types of chromosomes, X and Y. Females have XX. That is called homotypic. Homotypic. That is similar type. Now you see, the genes on these two chromosomes help in distinguishing between males and females on the basis of the sex organs, the secondary sexual characters and other features. So this is the classification of chromosome on the basis of their functions. Now there is another type of classification, second, that is called on the basis of position of centromere. Now you see that the centromeres are not present at same position in all the chromosomes. Now in this part you see, that is, the centromere may be exactly in the middle or slightly away from the center or near the end. So on that basis we classify the chromosomes on this part. Like you see, suppose you take this one. If the centromere is exactly in the middle, such that the two arms, this part and this part are called arms. Two parts are of the same size, then it is called metacentric. Metacentric. Second, if the centromere is slightly away from the center, such that one arm is longer and another arm is shorter, like this is one arm, this is another arm, then this is called submetacentric. Submetacentric. Similarly, if the centromere is near one end, such that the one arm is much shorter, another arm is much longer, then it is called acrocentric. Acrocentric. And then there is the last one, this one. That is, the centromere is at one end directly, and then it is called telocentric. Telo means end, so telocentric. Now, in prokaryotic cell, since the chromosome is single-stranded, so there is no position of centromere, there is no centromere, then it is called acentric, that is without centromere. So we have classified chromosomes on two bases, that is on the basis of function and on the basis of on the basis of position of centromere. Now, as we know that we have chromosomes in pair, like 23 pair. Normal cell in our body is diploid. Diploid means having paired set of chromosome. The reason is that through the gametes at the time of fertilization, one set of chromosome come from either of the parent. Like a sperm carry 23 chromosome, ovum carry 23 chromosome, and they together make 46, that is 23 pair. So we have to study here a term called homologous chromosomes. Homologous chromosome. The pair of chromosome of same shape, size and genetic location. Same shape, size and genetic location, one coming from either of the parents is called homologous pair of chromosome. Like you see, suppose there is a sperm. There are 23 chromosomes. Now, here there is ovum. Ovum also has 23 chromosomes. Now, when fertilization takes place and zygote is formed, then there are 46 chromosomes. That is 23 pairs. In each pair, suppose we take this type of chromosome. This type of chromosome. Then this suppose comes from father side, this comes from mother side. They have the same position of centromere, and suppose if on this chromosome the gene for length is located, then at the same position from the mother side there must be the gene for length character, that may be capital T or a small t. Here it may be capital T, small t, but the character position of the gene for a particular character is the same. So such a pair of chromosome is known as homologous pair of chromosome. Clear? Now, students, we have discussed different types. Now, one more term comes in this part to study, that is called karyotype. What is it? Karyotype. Karyotype is the photograph or picture taken to arrange the chromosomes on the basis of their shape, size in order to study is called karyotype. Any photograph taking or representing the arranged in same shape, size or in the order of shape, size and genetic location is known as karyotype. Now, so these were the different basic informations from the chromosome or structure of chromosome.